Okay, good evening, friends. So in the last class, in the last two classes, we discussed about the photosynthetic pathway. The other day, we discussed about the light phase of the photosynthesis, wherein we discussed about the structure of chloroplast, thylakoids, and various multiprotein complexes that are embedded in the thylakoid membrane. And they were photosystem 2, cytochrome BF complex, photosystem 1, and the last one was ATP synthase. And at the end of light phase, we have seen two important end products obtained. They are ATP and NADPH. These two are called assimilatory power. And of course, in the yesterday's class, we discussed how this assimilatory power is used in reducing carbon dioxide into a carbohydrate. Now, based on the first stable product that formed in the dark phase, where all the reactions of dark phase or carbon dioxide reduction phase occur in the stroma part. Grana reactions occur, light reactions occur in grana, whereas the dark reactions or carbon dioxide reduction occurs in stroma, we have seen. So in the stroma, we have seen yesterday, based on the nature of the carbon dioxide acceptor and the first stable product formed, the carbon reduction pathway is primarily of 3 kinds. Based on that, we have seen, we have classified plants into 3 groups. One is C3 plants, the other was C4 plants, and the last category was CAM plants. C3 plants, majority of the plants are C3, where the first stable product formed is a 3-carbon compound by name phosphoglyceric acid. Now here, the carbon dioxide acceptor is RUBP, ribulose bisphosphate 1,5 bisphosphate. And in the second category, of course, very few, maybe 2 to 3% of the angiospermic plants exhibit the C4 pathway. C4 pathway is so called because the first stable product formed is a C4 carbon compound called oxaloacetic acid or aspartic acid or malic acid. So hence, those plants are called C4 plants. Now, C4 plants, primarily, they belong to monocots, especially sorghum, maize, sugarcane, you come across. But of course, apart from this, even certain dicotyledonous members do exhibit C4 pathway, like members of our tribulus, a member of Zygophilase. Now, C3 plants, the cyclic pathway starting from RUBP to regeneration of RUBP. As so many reactions are occurring, so many conversions and reconversions are occurring, so many breakings, so many additions are taking place. So ultimately, it is regenerating or it is reaching the starting point. You're starting from RUBP after so many interconversions. Again, this RUBP is regenerated, and ultimately at the end of this, you see formation of one glucose molecule. If RUBP cycle or C3 cycle takes 6 rounds, then there is a formation of one glucose molecule. So this detailed discussion or detailed biochemical reactions were elucidated in detail by Melvin Calvin and his friends. Hence, this cycle is called C3 cycle, I told you yesterday. Now, C4 cycle, of course, till 1964, people thought that all plants do photosynthesis through Calvin pathway only, but it was Korschak, Hartenberg, Hatch and Slack. They were the people mostly from Australia. They worked on the method of carbon reduction in sugarcane, and they found there is an alternate pathway of carbon dioxide reduction or carbon dioxide fixation, and hence the C4 pathway is called Hatch and Slack pathway. And third, I told you, is what is called, so here in C3, C4 pathway, PEP or phosphoenol pyruvate is the a 3-carbon compound is the carbon dioxide acceptor. By accepting a carbon dioxide molecule, C3 acid, that is phosphoenol pyruvate, changes to a C4 acid. This is called beta oxidation, beta carboxylation, and the first stable product is a 4-carbon compound oxaloacetic acid. So these plants are called C4 plants, and a third group, only very few, especially plants belonging to Crassulaceae. So this was found in Crassulaceae, wherein the both C3 and C4 cycle. Say C4 pathway or C4 plants, two types of pathways occur. In C3 plants, only C3 cycle, but in C4 plants, in mesophyll, you see C4 pathway. In bundle sheath cells, C4 pathway, C3 pathway. So C3 in bundle sheath, C4 in mesophyll. So C4 plants show two methods of carbon dioxide fixation, hence they are more efficient than C3 plants. Now, in C4 the CAM pathway, all the reactions are similar, very similar to C4 pathway, but the difference is C4 cycles, in C4 plants, the C4 and C3 are spatially separated. That is, C4 occurs in mesophyll cells and C3 occurs in bundle sheath cells. But unlike C4 plants, in CAM plants, both the cycles, that is C4 cycle, C3 cycle, occur within the same cell, but different times, temporally. There is a time separation, whereas in C4 plants, there is a spatial separation. So here, crassulation plants like our bryophyllum, which are succulents, which are xerophytes, generally there, they close their stomata during daytime to conserve, to check transpiration. So during nighttime, they open their stomata. So when the stomata open, then carbon dioxide in the form of bicarbonate ion enters, and this bicarbonate ion fuses with PEP, and of course, same pathway. So here, the oxaloacetic acid and malic acid are stored in the vacuole during nighttime, and when it becomes day, then there is they and there the oxaloacetic acid, breaks into malate, and of course this malate decarboxylation, dehydrogenation, of course finally starch, and whatever the carbon dioxide that is released will be fixed by RUBP. So like this, during daytime C3 cycle, during nighttime C4 cycle. So like this, even crassulation acid metabolism plants also exhibit two cycles. Of course, these we have studied. In addition to that, we have also studied about photorespiration is nothing but light-induced respiration. So normal respiration occurs during night and day. But unlike that, normal respiration, certain plants, especially C3 plants, show photorespiration, a light-induced respiration. So when there is a lot of light, or when the intensity of light is more, when the temperature is more, when oxygen concentration is more, when there is a fall in the carbon dioxide concentration, under those conditions, RUBP carboxylase, instead of doing carboxylation, it does what we call oxidation. So RUBP oxidase. So thereby it breaks the 5-carbon RUBP, ribulose 1,5 bisphosphate. So through a series of biochemical reactions, ultimately PEP is, phosphoenol pyruvate is regenerated. Here, with the breaking of RUBP, you get a 3-carbon PEP and a 2-carbon glycolic acid. As the glycolic acid is the compound that makes so many cycles are undergo so many biochemical modifications. Hence, this cycle is also called C2 cycle, and it is also called photorespiration. And here, the photorespiration involves not one cell organelle. Generally, the normal respiration, for the normal respiration to take place, only mitochondria takes part. But unlike that, the photorespiration involves a combined effort of three cell organelles, that is chloroplast, peroxisome, and the mitochondria. So a sequential biochemical reactions take place, and ultimately, in each cycle, there is a decarboxylation, and this carbon dioxide goes waste. So hence, through photorespiration, there is something like 25% of wastage of energy. So it is a wasteful process. So we discussed all these things in yesterday, because why I am repeating this, the photosynthesis is very, very important as far as our examination is concerned. You will surely get one essay from this and one or two, short notes, like differences between C3, C4 plants, or various internal and external factors regulating or this photosynthesis. They may ask, or they may ask, give C3 cycle, or they may ask photorespiration, they may ask, or light reaction, they may ask, like this. So this is the most important topic. Hence, I used some time of this today's class for the yesterday's revision. Now, in today's class, we'll discuss about the translocation of solutes. The translocation. In translocation, the tissues that are involved in translocation are conducting tissues. What are the conducting tissues? The conducting tissues make what we call vascular bundles. Each vascular bundle has the two important, tissues. They are rather complex tissues made of different types of elements. They are nothing but your xylem and phloem. Of course, you read about in detail, about these tissues in earlier semesters. Now, xylem is primarily concerned with transport of water. It is, of course, we have in plant-water relations also, I told you how ascent of sap takes place, and in that we discussed. So Dixon's method of ascent of sap we discussed. So xylem is primarily concerned with transport of water and mineral salts, and the transportation of photosynthetes. So say we discussed in detail about photosynthesis. At the end of the photosynthesis, what is the end product? That is nothing but sugar. Sugar ultimately stored in the form of starch. Now this sugar or starch is produced in leaf. So this is the site of source. So food is manufactured in leaf. Now leaf uses some of its photosynthetes for its survival, for its growth, and the rest of the photosynthete is translocated to the areas where they are reserved or preserved. So where this reservation or preservation or storage takes place, there are various storage organs in plants. Different crops, different storage parts. Say, for example, it is a palak or leafy vegetables. There, all the photosynthetes are located in the leaf itself. If it is a flowering, that is, for example, cauliflower, it gets translocated to flower, from leaf to flower. If it is seed, say for example, all our pulses, all our cereals, rice, wheat, barley, rye, oat, maize, jowar, In all these things, the important part is, which is the important one? Seed is the important part. So if it is mango, it is watermelon, it is muskmelon, it is orange. In all these things, apple. In all things, tomato, or your brinjal, grapes. In all these things, which is the economically important part? It is the fruit. Now there are certain crops, tuberous crops. It is a potato, sweet potato, radish, beetroot. In all these things, which is the part where the food material is stored? These are tubers. They are subterranean within the soil. So the leaves are aerial. They are in the air above the ground. Whereas tuberous crops, the storage part is within the soil. So whatever the food that is manufactured in the leaf has to be translocated to the storage areas. Maybe a tuber, maybe a fruit, maybe a flower, maybe a seed. So whatever, depending on its economic important part, from the manufacturing part, that is the leaf, it has to be ultimately translocated to the part where it is stored. So this translocation of food materials, maybe your sugars or starch, from the source, that is leaf, to the storage organ, otherwise called sink. So the translocation from source to sink, depending on if it is a herb, the distance is less. If it is a big tree, distance will be more. If it is in tuber. So always, whatever the case may be, the food has to be transmitted from source to sink. So this translocation of food materials occurs through phloem. It occurs through phloem. So this translocation of food materials from the leaf to the storage organs or from the source to the sink is called translocation of food materials or translocation of solutes. Now, for this, how actually this takes place? Which tissue is involved? This was worked out by, Hurtig. So Hurtig performed what we call ringing experiment or girdling experiment. In this, what did he do? He removed the bark around the stem or a branch in the form of a ring. Hence, it is called ringing. Ringing experiments are, as it is in the form of a girdle, hence it is called girdling experiments. So when you remove the bark, what do you find? If it is a perennial, if it is a herb or a shrub, if it is a tree or a shrub, you see bark. If we remove the bark, what do you find inside? Hard material, that is wood. Wood is nothing but xylem. The superficial part, the delicate superficial with liquids, maybe latex, maybe watery substance, that bark is nothing but phloem. The superficial part is, the superficial delicate part is phloem, whereas the deep interior central cylinder, which is very hard, is nothing but xylem, otherwise called wood. So by doing ringing experiments, Hurtig observed that when the material was removed in the form of a ring, the food that manufactured in the upper part or in the leaves translocated downwards, and because the bark was removed, it accumulated just above the ring. Hence, that upper part became bulging. So this bulging experiment, ringing experiment, that bulging is because of the storage, accumulation of food materials. Because of the accumulation, that upper part became bulged. This is what we call ringing or girdling experiment. So through this ringing girdling experiments, we came to conclusion that the bulging is because of the accumulation of substances that traveled from leaf to the lower part just above the girdle, and because the lower part was cut, there is no scope for the movement. The connection got removed in the form of a girdle or in the form of ring. Hence, it got stored. So through these girdling and ringing experiments, we came to conclusion that it is not the xylem, it is the phloem that is involved in the translocation of various food materials or solutes. Now structure of the phloem you studied in anatomy. Now the phloem consists of 5 important elements. One is called sieve tubes, companion cells, phloem parenchyma, and phloem fibers. So of them, so generally the fibers are non-living, the other 3 are living. The sieve tubes are living. Initially, young sieve tubes are nucleate. As the sieve matures, it becomes anucleate without nucleus. But the companion cell, which is adjoining the sieve, has definite nucleus. It is living. And the parenchymatous cells that are associated with the sieve elements are also living. So the sieve element, it is like this, narrow in the center and expanding at the tips. And here you will find at both the ends, you find sieve. This sieve area has so many holes. Through that, from one sieve element to other sieve element, there is a streaming of protoplasm. So the movement of the cytoplasm from one cell to other cell occurs through these sieve elements, these pores. This pores is called sieve plate. That plate that divides the 2 subsequent sieves has a sieve-like area called sieve plate with so many holes. Through them only one sieve is connected to the other. So this connectivity, the cytoplasm or protoplasm moves in the form of what we call through plasmodesmata. Through plasmodesmatal connections from one sieve element to other sieve element, or from sieve tube to companion cell, or from sieve to even your what we call parenchymatous cell. There is a movement. So mainly it occurs from one sieve to the other. So sieve elements are primarily involved in conduction of food materials. So so many people worked how actually this movement or translocation of sugars or photosynthetic or assimilates. Photosynthetic end products are translocated from source to sink. So there are 2 important theories to explain this long distance transport of food materials. One is what we call, one is through protoplasmic streaming. So one tells that cytoplasmic streaming is one. So generally you'll be surprised, the movement of food materials through sieve elements, not always longitudinal, but sometimes even in a transverse way, in a sideways also, it takes place. So upward, downward, sideways. So radial waves, tangential waves, even vertical waves in all directions. So not in one direction, but either ways. Upward towards upward and also towards downward. Bidirectional movement of these solutes has been observed through various experiments conducted by various people, various scientists. Now it is clear that the transport in the phloem is bidirectional. It's not unidirectional. But earlier theories, especially the cytoplasmic streaming, tells that here cytoplasmic streaming is nothing but movement of the cytoplasm around the vacuole. It is what we call cyclosis. So it moves around. This what we call cyclosis. So especially Hugo de Vries. Hugo de Vries in 1885 put forth this cytoplasmic streaming as the method for the translocation of the cytoplasm from one cell to the other cell, from one area, from one part to the other part. But now this cytoplasm cytoplasmic streaming, it shows that it tells that only unidirectional, but this through using by injecting viruses, etc., they have worked. They have even by radiography. So this isotopes, radioisotopes, they determined the actual movement, how actually the cytoplasm makes movement. So by using various radioactive elements and also by using viruses, they found how actually the long distance transportation occurs. Now, this long distance movement occurs, this cytoplasmic streaming may holds good for maybe to herbs, herbaceous, but long shrubs and trees, it is very difficult to subscribe to only cytoplasm streaming for this long distance translocation. So, to explain in a better way the actual mode of translocation of these food materials, in 1930, a German, the biologist, especially German, by name Munch, put forth his theory, popularly called Munch hypothesis or mass flow hypothesis. Munch hypothesis or mass flow hypothesis says that along with the translocating materials, along with that, even the food materials also get translocated in en masse way. In en masse way, they get translocated. Hence, it is called mass flow, otherwise called pressure flow hypothesis or mass flow hypothesis. Now, he says that the food is manufactured in the leaf, and with the dissolution of, with the formation of sugars, their osmotic potential falls, and then water, through water potential from the surrounding cells, from parenchymatous cells, and even to certain extent through xylem also, water makes an entry into the phloem cells or into the sieve tubes, and with this entry, positive hydrostatic pressure is created in the sieve tube elements. Under this tension or under this turgor pressure, the fluids along with its dissolved sugars get translocated from the source to sink. So from the leaves to the area where they are stored. So to explain this, even for example, this toddy. Toddy, generally in case of Phoenix sylvestris or Phoenix dactylifera, Borassus flabellifer, where toddy, generally toddy is extracted in the form of sap. That sap is nothing but very sweet, consisting of sugars. So here, water plus sugars, food that is, if you make an incision in the phloem, you will see the water with solutes oozing. So ultimately, in 12 hours or so, you will see some 5 to 10 liters of this toddy getting collected. So that toddy is nothing but long distance translocation of food. So from leaf to the incision areas, this food along with the liquids get translocated and get collected by these toddy tappers. So to explain this long distance mode of translocation, he explained by using a physical model, Munch hypothesis. For this, to explain this, he has taken, two closed osmometers made of glass and also two troughs, water troughs, the glass troughs, but taken, and one osmometer A and other osmometer B. In A osmometer, so the osmometer is nothing but the glass mouth is covered by semi-permeable membrane. Then it becomes an osmometer. So semi-permeable membrane allows only certain particles to pass through it. It doesn't allow. It has discretion, which solute has to be passed through, which solute's movement has to be, uh, you know, do not allow. So which particles are to be allowed, which are to be blocked, will be determined. Hence, it is called semi-permeable. It permits only certain materials to pass through it. Hence, it is called a semi-permeable membrane. So here, this consists of what we call two, troughs, and say see, for example, this is this is a Munch apparatus. This is what we call, these two are osmometers, and this is a class stuff, and both of them are interconnected. And this osmometer is called A, and the other is called B. And in A, generally, the solute concentration is more. That is, the osmotic potential is negative here, and in other dilute. So here the solute particles are less. So compared to A, here the water potential is more. So because of these two, A and B are differing in their concentration of solutes. Now these two are placed in glass tubs, and these in turn they are connected by the water. And because here in A osmometer, more sugars are there, and osmotic potential is, or water potential is negative. Hence, the water which is whose water potential is more make an entry into heat. So under this, high hydrostatic pressure or turgid pressure develops. Under this, the water moves along with its solutes and enters into B. Now the B, on receiving this, now here also, because when compared to A, this is dilute, but still when compared to the water here, it is also, its water potential is negative. Hence here also water enters. And because from this B, always solutes will be extracted. That is storage. They'll be continually passing to storage. So always this will be drawn. So there will be a deficiency of, solutes here. So continuously from A to B, there is a movement of, solutes along with water. So this is called source. This is called sink. Now this A is taken as what we call leaf, and B is taken as what we call, this B is the sink, and this is the phloem, that is sieve elements, And these troughs, we can equal them to what we call xylem element. Maybe your tracheids and trachea. So always from tracheids and trachea, water may make an entry and develops hydrostatic pressure. Under this, along with water, the solid particles move into the sink. From sink, continuously they'll be drawn into the sink, and there is always a deficiency. So from source to sink, there is a continuous translocation. So according to Munch, he says that only deficiency is that Munch, of course, it explains well the translocation. But the thing is, he said that this mass flow or pressure flow hypothesis, mass flow hypothesis, does not need any energy. It is passive mass flow or passive flow. But recent, works by various scientists proved that the temperature, even hormones, and even ionic absorption have a bearing in the translocation of solutes. So if the temperature is more, then the translocation gets enhanced. And when certain inhibitors were used, now they checked the rate of translocation. This proves that energy is needed for translocation. So in addition to this passive movement, even active movement is also involved. That is, energy regulated movement is also possible there. So according to latest information, though it is passive, but even energy requirement is also needed. When sufficient energy is provided, the translocation gets enhanced. When the under bright sunlight or under increased temperature, the rate of translocation has been proved to be accelerated. It has been proved to be very accelerated. So these facts tell. So the Munch hypothesis, of course, tells us, it gives us, it tells us, conveniently, and more people agree to the mass flow or pressure flow hypothesis, but there are certain demerits or criticisms. So cytoplasm in the sieve, one criticism is that cytoplasm in the sieve tubes is not completely dilute, it is rather viscous. When it is viscous, generally viscous substances cannot move freely through sieve areas. This is so the viscous nature of the solutes, becomes an impediment for their free movement. But there are certain natural conditions, which the protoplasm, the cytoplasm do not always remain viscous, but with the entry of water from the auxiliary elements, it becomes normal, and whatever the impediments in the form of what we call tallows plaques that block this sieve areas get removed in due course, and there is a this continuous movement of translocation from source to sink happens. Now there is also one more thing is that transportation all solutes should be, according to Munch, unidirectional. But however, experiments reveal that substances do not always move unidirectional, but even bidirectional movement has been established. So this bidirectional movement has been proved, convincingly, that in phloem, the movement not always unidirectional, but even bidirectional movement is also possible. And there are certain, substances that regulate the rate of movement. Now, especially there are certain substances, especially the boron. The boron concentration has been found to be one of the important factors for the translocation of solutes or the sugars. If plant suffers from boron deficiency, the translocation gets affected. So also the growth hormone 2,4-D is known to regulate the phosphorus movement. So another experiments, especially with radioisotopes, it revealed that solutes translocate not unidirectional but bidirectional movement is being done, and this is for the faster movement. Now, recent you know uh, recent experiments revealed that there is what we call always uh, sugar transport is linked to proton movement and also ATP involvement. So this movement of protons and sugars, they go together. This is what we call symport. Two molecules, that is one ion and the sucrose, are going together in one direction. Same, both in one direction. This is what we call a symport. Now, the protons for the symport come from ATP. So when ATP in the form of phosphatase becomes ADP, protons, we have seen, in light reactions, we have seen ATP synthase, wherein ATP synthase for the movement of 4 protons, 1 ATP synthesis takes place. This is what we call coupling. And ATP synthase we have seen. So here protons are needed, or proton motive force is used for a phosphorylation or for conversion of ADP into ATP. So here, if the plant needs protons, here the ATP becomes ADP and protons are released, are made available, and these available protons are utilized by the sucrose and make movement together. This what we call a symport movement. Now, see for example how actually this movement of long translocation occurs. See for example, this long translocation, if you observe here, you can see for example, this is sugar molecule and these are plasmodesmata, and of course, from one cell to other cell, you can see entry. Now, these are all mesophyll cells. From mesophyll, it is entering into what we call bundle sheath or endodermal cells. From here, you can see this phloem parenchyma cells. From phloem parenchyma, you will see the companion cell, and ultimately they are seen entering into sieve elements. So there is a movement from what we call from apoplast to symplast. So for apoplast movement, no energy needed, but for the symplast movement of sucrose, energy is needed. So hence the movement with the involvement of energy is called an active movement, and without energy is called a passive. So passive translocation and active translocation. So by utilizing ATP, the translocation has been found to be very, very fast. So this is how. So even role of hormones, it has been found that the mobilization of solutes from storage to sink in the newly developing sinks is considered to be under control of growth regulators. So many growth regulators are known to help in the faster movement or the faster translocation of solutes from source to sink. Now, finally, this what we call harvest index. Now, this photosynthate, what we call synthesis of photosynthates or glucose, and especially the starch has taken place, and this starch has to be translocated to zinc. Already I told you. Now the zinc has to be uh, uh, the translocation should be zinc, and zinc should be an useful part. See, for example, in certain crops, they show what we call luxuriant vegetative growth. If a plant grows, whatever, take any example, if it shows excessive growth, so what is happening? Too much leaves, too much vegetative growth. So plant, see, for example, if it grows very lengthy, the cob will very small. The inflorescence are the what we call the seeds produced will be small and less number. The panicle, what we call the spike and the spikelets, what we call the spikes and spikelets, or the cob will be very small. Even the yield will be very small. So hence nowadays, through breeding techniques, what the breeders are doing, breeders are working for shorter duration, short stature ones. Short statures ones, here the usage of photosynthate for vegetative growth has to be restricted, and whatever the photosynthate that has been produced, it has to be translocated to the sink so that yield gets enhanced. This is what we call the harvest index, which tells us that for calculating or say harvest index, the economic yield by biological yield into 100 gives us harvest index. So all the scientists or all the agriculture is nowadays trying to reduce or to restrict the movement of photosynthate to vegetative parts, and diverting as much photosynthate as possible to the sink area, so that your agricultural yield gets enhanced, or your harvest index gets enhanced. So this is long distance translocation or translocation of solutes. Now the 5 minutes is there for us for discussion. You can raise your doubts, not only regarding today's class, but also regarding previous classes also. If you have any doubt, you can raise your doubt. So now with today's class, we have completed almost we have 12 classes allotted for us, and with today's class we have completed 6 units, and we have 6 more classes, and of course, and in the leftover 6 classes we'll discuss about respiration, nitrogen metabolism, plant growth regulators, and we'll discuss in ecology, ecosystem, ecological succession, and also environmental pollution. So 50% of the syllabus we have covered in the last 6 classes, covering water relations, mineral nutrition, enzymes, photosynthesis, and translocation of solutes. In the leftover, but of course, as far as examination is concerned, photosynthesis is very, very important for you. Hence, I try to use maximum time to make you understand the important biological or biochemical or primary biochemical reaction, that is photosynthesis. I think We have classes on next Friday, Saturday, and Sunday. In the next three classes, we'll discuss about respiration and nitrogen metabolism and plant growth regulators. And as you know, this respiration is just reverse to what we discussed in photosynthesis. Photosynthesis is an anabolic process wherein simpler molecules in the presence of sunlight, by using the chlorophyll pigment, energy-rich carbohydrates are produced. This is called anabolic process, and opposite to that anabolic process is catabolism or catabolic process, wherein energy-rich products, especially the glucose gets broken into simpler molecules, that is carbon dioxide, water, and ultimately there is a release of tremendous amount of energy. So this is respiration is an exothermic reaction, whereas photosynthesis is an endergonic or endothermic reaction. So we will discuss about the respiration, and the important organelle that is involved in respiration is our mitochondria, as chloroplast is for photosynthesis and mitochondria is for respiration. Hence, the mitochondria are called powerhouses of the cell. So we will discuss about that in the next class. Now, any doubt? So I gave you 5 minutes for you to express your doubts, but none of you are raising any doubts. So there are two conditions under which a person do not raise any doubt. One is if you understand everything, or if you don't understand anything. But I think you don't fall under any of the categories, not completely understanding the things and not getting anything. This is not the case. But anyway, I thank you for your patient hearing and hope to meet you in the next class. Until then, bye.